In this video segment, we will be studying meiosis. The reason that cells go through meiosis is to produce gametes for sexual reproduction. This is important because it is one way to get genetic variation. In organisms that use sexual reproduction, DNA comes from each parent. So this means that parents will each contribute one chromosome of each type to their offspring. For this to work, the daughter cells of meiosis must have half the amount of DNA as a parent cell. There are three major ways to get genetic variation for sexual reproduction. The first one is random fertilization. Which egg and which sperm fuse together during fertilization is actually a random process. The second way is independent assortment. During metaphase I and metaphase II, chromosomes line up independently and randomly at the metaphase plate or the cell equator. This random assortment, also known as independent assortment, provides genetic variation. The third way is crossing over. During prophase I, pieces of chromosomes will actually break off and exchange places. This means that pieces of maternal DNA are exchanged for pieces of paternal DNA, giving us even more genetic variation and possibilities in the offspring. One particularly important concept when talking about meiosis is ploidy. Ploidy is the number of chromosomes found inside of a cell. We are going to talk about two types of ploidy, haploid and diploid. Let us say that there's a cell that has three types of chromosomes. The first chromosome is large. We'll call it number 1. The second chromosome is squiggly. We'll call it number 2. And the third chromosome is small. We'll call it number 3. If a cell has only one of each type of chromosome, then it is haploid. And we abbreviate haploid with a lowercase n. If the cell has the same three types of chromosomes, but it has two of each type, two large, two squiggly, and two small, we say that cell is diploid. Diploid is abbreviated with a 2n. Here is an example of a karyotype from a haploid human cell. Humans have 23 types of chromosomes total. Most of those chromosomes are numbered 1 through 22. Those are the autosomes. The last type of chromosome is our sex chromosomes. They are not numbered, they're just called sex chromosomes. The only type of cell in humans that are haploid are our gametes. So those are the egg and sperm cells. A cell that is diploid has two of each type of chromosome. So here in this karyotype below, we see that there are two of each type of chromosome. Again, the chromosomes are numbered. We see those numbers down on the bottom with sex chromosomes at the end. So diploid is two of each type of chromosome. So here's a nicer picture of a human karyotype. A karyotype is just a picture of all the chromosomes from a cell. So this is a human karyotype. We can see that there are 23 types of chromosomes found in this cell. Again, most of these are numbered. There are 22 autosomal chromosomes and 2 sex chromosomes at the end. Most human cells are diploid, having 2 of each type of chromosome. When we talk about chromosomes of the same type, we are talking about homologous chromosomes.
These homologous chromosomes are the same size, have the same banding patterns, and have the centromere in the same location. They're not only the same in structure, but they also have the same genes. Here's another karyotype. In this karyotype, we see that the bands have been colored. Each homologous pair has the same bands, the centromeres in the same location, and are roughly the same size. The exception is the sex chromosomes. We have discussed how male sex chromosomes, the Y chromosome, is much smaller in size than the X chromosomes. Remember, after replication, each chromosome is made of two sister chromatids held together at the centromere. These homologous pairs will eventually pair up together to create a structure called a tetrad. We've learned previously that the cells go through two major parts or steps of their cell life or cell cycle. The first is interphase. The second is cell division, which is much shorter than interphase. We see that no matter if a cell is going through mitosis or meiosis, they will always be in interphase for most of their life. The interphase has three important parts, G1, S, and G2. G1, or gap 1, is the phase where the cell goes through normal everyday processes. S phase stands for synthesis. This is when DNA is replicated. G2 phase is getting the cell ready for cell division. Meiosis has two rounds of cell division, Meiosis I and Meiosis II. The steps of meiosis are very similar to those of mitosis. We will differentiate between the different steps by using numbers. So during meiosis I, all the phases or steps will have a 1 behind them. So for example, prophase I, metaphase I, anaphase I, telophase I. During meiosis II, all the steps will have a 2 after them. Prophase II, metaphase II, anaphase II, telophase II. Like with mitosis, the best way to learn meiosis is to sketch the steps of meiosis and to write down what's going on. I'm going to walk you through how to do that. So during meiosis I, we're going to see that this step focuses on separating out the homologous pairs into different cells. So a quick review. During these sketches, I will draw the centrosomes as small boxes. The spindles will be the lines coming off of those. We know that chromosomes come in many shapes and sizes. For example, a single chromosome can be made of one chromatid. During replication, that chromatid will be copied. This will give us one chromosome made of two chromatids. These chromatids are held together by a structure in the middle called the centromere. As long as these two chromatids are held together as one structure, they are a single chromosome. In meiosis, we take these single chromosomes that are made of two chromatids and pair them with the homologous chromosomes, or the chromosome of the same type. We have one chromosome that originally came from your father, the paternal chromosome, and one chromosome that originally came from your mother, the maternal chromosome. This large structure is called a tetrad. Tetra stands for the number 4. There are 4 chromatids in a tetrad. Tetrads form so that crossing over can occur. Crossing over happens during prophase I of meiosis. During crossing over, we see pieces of non-sister chromatids physically breaking off and changing places. This is a form of genetic recombination, which gives us new variations in the offspring. So let's start with interphase. During this time, our cell will have a nuclear envelope. The DNA will be loose and stringy in the form of chromatin. During this time, the DNA will replicate during the S phase of interphase. In G2, it will then prepare and get ready for meiosis. For this example, we're going to say that this cell has 3 types of chromosomes. We can sketch a little karyotype of this. After replication, our cell will have 2 of each type of chromosome. So large chromosome, small chromosome, medium chromosome. This means that the cell is diploid. The cell will have a total of 6 chromosomes at the beginning. The first step of meiosis I is prophase I. This is a very busy step that will have 6 different events going on. During this time, we're going to see that the cell's nuclear membrane is going to be dissolving. The chromosomes will be condensing and forming tetrads as they go. Crossing over will also happen during prophase I. As all of this goes on in the nucleus, our centrosomes will be forming and the centrosomes will be moving towards the poles of the cell. We can see at this stage that the cell is still diploid. The second step of meiosis I is metaphase I. During this stage, we're going to see that the homologous pair will be lining up along the center of our cell, so along that metaphase plate. This is a random process called independent assortment, and this helps give extra genetic variation in the offspring. During metaphase, we also see that those centrosomes have reached the poles and that they have produced spindles long enough to attach to each of the tetrads. At the end of this phase, the cell is still diploid. The next step of meiosis I is anaphase I. During this stage, we know that the spindles are going to shorten, pulling apart our tetrads so that one chromosome from each tetrad goes to either side of the cell. We saw during mitosis that telophase I often occurs at the same time as cytokinesis, even though these are 2 very different things. During cytokinesis, we will see the overall cell start to pinch, creating a cleavage furrow that will divide the cell into 2 cells. The cytoplasm will be split evenly. During telophase I, we will see that the nuclei of each cell will start to reform. The DNA will become loose and stringy. And the centrioles and spindles disappear. At this point in meiosis, the homologous chromosomes have separated, meaning we are in the process of forming 2 haploid cells. At the end of meiosis I, we have formed 2 daughter cells. Each of these daughter cells has 1 of each type of chromosome, meaning that they are now haploid cells. If we zoom in on the nucleus of each, we will also see that there are unique sets of DNA on each side. This is because crossing over has occurred. In other words, we now have two unique cells. At the end of meiosis I, cells go through a brief period of rest called interkinesis. Once the cell is finished with interkinesis, both daughter cells enter into meiosis II. During this second round of division, the cells will focus on separating out the sister chromatids into separate cells. Prophase II is the first step of meiosis II. During this stage, both daughter cells will be doing the same things. The first part of prophase II is that the nuclear envelopes of both cells will begin to dissolve. Next, the DNA will start to condense, causing the thick X's of chromosomes to become visible. During this time, we also see that the centrosomes will be forming spindle fibers. As the spindles form, the centrosomes will also be migrating to each pole. The next step is metaphase II. During this phase, the chromosomes of each cell will line up along the cell equator. During this time, we also see that the centrosomes have reached the poles. The spindle fibers have lengthened and are now attaching to the centromere of each chromosome. The next step of meiosis II is anaphase II. During this time, we see that the spindles will shorten. This will cause the sister chromatids to separate and be ripped apart. The sister chromatids will move to opposite ends of the cell. The last steps are telophase II and cytokinesis. Again, during cytokinesis, we will see the pinching of the cell, the cleavage furrow forming. During telophase II, we will see the nuclear membranes reforming. The DNA will become loose and stringy, becoming the form of chromatin again. This will occur for both of our daughter cells. We are now in the process of forming 4 haploid daughter cells. At the end of meiosis, we will now have 4 daughter cells. Each of these daughter cells will be haploid. This means that each cell will have only one of each type of chromosome in the nucleus. We will also see that each of these cells has a unique combination of DNA inside of the cell. If we zoom in, we can see that because crossing over has occurred, each of these chromosomes is a little bit different than the original parent chromosome. Once these gametes are done developing, they're ready for sexual reproduction. A haploid egg cell can combine with a haploid sperm cell to form a diploid cell called a zygote. The zygote will then go through mitosis, growing and growing until it becomes an embryo, a fetus, and eventually a baby. All of the cells in this baby will be diploid. The last thing I want to do during this video is to compare and contrast the process of mitosis and meiosis. We can compare and contrast the end products of these two. Mitosis has 2 daughter cells at the end of 1 round of division. These daughter cells are exact copies of each other. During meiosis, we see that there are 2 rounds of division, resulting in 4 daughter cells, each that are unique and different from each other, different from the parent cell. We also know that mitosis, because it is making an exact copy, will start with a diploid cell and end with a diploid cell. Meiosis is starting with a diploid cell and ending with haploid cells. The purpose of these 2 cell divisions is very different as well. We learned previously that mitosis is used for growth, repair, and asexual reproduction. Meiosis is only used for sexual reproduction. We also know that the steps of meiosis and mitosis differ. Meiosis 1 focuses on separating out chromosomes that are homologous into separate cells. This does not happen during mitosis. Meiosis produces tetrads that then have crossing over occur. Again, those things do not happen during mitosis. Meiosis 2 focuses on separating sister chromatids into separate cells. This is very similar to mitosis. During mitosis, sister chromatids are also separated out into separate cells. During this video, we have learned about ploidy and about the process of meiosis. Be sure that you can compare and contrast the process of mitosis and meiosis, and that you can fully describe the steps of meiosis and the end products.